we're going to basically pick up on a slightly zoomed version of what we did last time, where we gave an overview of a lot of different kinds of viruses. And then actually, accidentally, we ended up focusing a lot on AAVs and lentis and a bit on rabies, and we're going to focus entirely on that. The first two, AAVs and lenti, follow under the same framework where they're basically vehicles for delivering genes. And because of that, they can be used for a large variety of applications, many, many, many ones. Whereas the rabies virus is a bit more unique in that, as far as I know, its main use is for basic science, is for transsynaptic tracing. So it has a much more limited scope. But it'll still follow the same format as before, where we're going to talk about it in terms of a little bit about the basics, the basic guts of the virus itself, and then talk about some common applications. You can see here on the side of the screen that it'll have basically what part of the talk we're in, just so you can orient yourself. And there's not going to be as smooth of a narrative transition between them. It'll feel just like a whole bunch of punches of different ways these things are used. But that's by design, because, you know, when you're looking for your own biological question, you're not really putting the tool in terms of this unified context of where it flows into. So it also doesn't super make sense to talk about tools in that way. It becomes more of like a random grab bag. And I've tried to organize it as logically as possible, but that is often not really possible. So with that, I'm going to step right in and start with the common viruses used, starting with the AAV. So the AAV is a very small genome of DNA, single-stranded, that has these inverted terminal repeats, ITRs. These types of repeat structures are common to a lot of different viruses. You'll see that the lentiviruses have LTI, long terminal repeats, whereas the rabies virus doesn't have something like that because it's very, very different. And this particular virus is much easier to discuss because it has only two genes, the rep and the cap, and they encode for 4 proteins each. The ones in black here are the ones that are required for the viral capsid assembly. The one in red are actually at the outside of the capsid, which actually form the structure that is the one that allow for viral entry. Now, there are certain issues about viral capsid structure and the symmetry that they have, which I kind of spend a lot of time drawing this uselessly because it turns out that it's easier if you just see with a little thing. So because of Corona, oh, and my camera just decided to really not like the focus on this. So, you know, it's an icosahedron, so it's 20 triangles, which I kind of cut together and put them in place. And you can see that there is an axis of symmetry, which is here, where the left side is symmetric to the right. There's an axis of symmetry, which is here, which is a 3-fold axis of symmetry. The previous one was a 2-fold, and this is the 5-fold axis of symmetry here. And I drew it like this so you can see that each facet of the thing has each of the axes of symmetry, meaning that if the virus is like bouncing around to infect something, each of the facets that has a certain functional property would be able to have a particular ability to bounce onto what it needs. In particular, the VP proteins that are required for the attachment to the cell through the heparan sulfate proteoglycan are right in the middle of the 3-fold axis of symmetry, where the VPs kind of have that little, a certain set of amino acids that are sticking out that seem to mediate that particular binding. I both just wanted to show off this and justify the time I spent on it. And so what people do when they grow AAVs and you buy them from Agene or from your core is that you can get them in a certain serotype. Serotypes are basically what is the rep cap that you're using to make your virus. And they always come from certain patient samples. They're named 1 through 9, and later ones are usually evolved from those samples. So if you get something, it used to be that you called it AAV 2-1. And that's because the AAV construct that you're using, which I'll show later that has your transgenes and whatever, are from backbone 2, from the AAV serotype 2, which is the mostly studied one in basic virology. But the cap proteins itself, the ones that are used to make the proteins that are in the outside of the capsid, are from 1. Recently, people stopped calling it like that, and they start just saying things like AAV1. because it's assumed that the genome is always going to be from 2. No one really changes that. So this is just what the cap gene is. And now they're also making chimeric versions that are mixtures of caps, say chimeric 2-1. And that is still the genome from 2, it's just that the cap protein has a mixture of them. And other serotypes also refer to just the cap gene, not the DNA inside the viral particle itself. That's because when you use this virus, you gut out the rep and cap and you put in a promoter of some sort and your transgene. And let's say you go to add gene and you can see something like this, and it has the promoter, the transgene, the untranslated region, which is usually added to increase expression, but is often not necessary for it, and sometimes even decreases it. And you need a poly-A sequence. And you often have the serotype here, and they're using now the new nomenclature where they just call it 1, and they just assume that, yeah, the genotype is of the genome is from 2. And you can note here that you can have all variety of transgenes, red fluorophores with a calcium sensor, with a peptide cutter, all sorts of whatever you need for your project as well. Now, when you make these viruses, so what you have is you have your transfer plasmid, which is just your ITRs with a promoter and the transgene that you want, all less than 4.8 kb, because that's the effective packaging size of AAVs. You can go over that, but you'll suffer a lot in terms of the titer you'll get. And you might even get truncated versions, which will just not work. And then you co-transfect that into 293 T cells with a separate promoter that actually produces the rep and cap proteins. This is why it's easy to swap out serotypes once you have your particular thing. And it's an adeno-associated virus. So it also needs these genes from the adenovirus in order for the virus to be constructed. You then collect the virus from either pellet or supernatant or a combination of those. There's a variety of different methods that I'm not going to talk about. And then you actually end up with your construct like this that is nicely packaged in an icosahedral capsid that has your cap, VP1, VP2, VP3 complement outside, which then mediate infection. Now, this is a DNA virus, so very usefully, you can actually make it Cre-inducible. So if you have the double inverted ORF system was actually developed, as far as I know, and using the first time at least extensively for AAVs. And I'm not going to describe the system in detail. You should look it up if you plan on using Cre-inducible things, because this is really the most effective way and leakless way to induce Cre-dependent activity, where you have incompatible Cre sites and a construct that's reversed in the reverse complement orientation that in the presence of Cre gets put in the right orientation. This is not entirely leakless though, because you do get some transcription and translation from the ITR, which often acts as a minimal promoter. You could also do this with other recombinases like the FLIP recombinase and these FRT sites here. That's not nearly as common or necessary though. Now, one particular note, this is important, and more of a historical at this point, but there used to be a difference between DIO, double inverted open reading flame, and FLEX, which I forget what it means. But nowadays, they're functionally the same. And note where the ATG starts, though. Like in all the DIO constructs, the ATG is within this transgene here, where it would normally start. Now, the problem is that some older variants of these viruses that were in the flex, named as flex, have an ATG right outside here, outside the promoter, meaning that you're always transcribing some crap and might be translating something, depending on how the construct is put together. And this is generally not what you'd want, but sometimes you do if you actually have a lot of leak from this other thing here. So this is just something to bear in mind. Here you see an example of a floxed allele that in the presence of the recombinase, you can see a lot of expression, in this case, just membrane GFP, and its absence, nearly nothing. Well, nothing you can see at the same exposure as this one here. And this is far better than other systems such as the lock-stop locks, which works reasonably well when inserted into genomes, like many reporter lines work that way. But for AAVs, this is by far the best way to do that. You can then combine these into intersectional AAVs, and intersectionality is very important as a concept on using viruses, because you can then look at different properties of your biological system and combine them to really just target something that you want. And in this case, the Diceroth lab have found ways to, using introns that have either the flip recombinases or the Cre recombinases to allow viruses that express in the presence of both Cre and flip or only one or the other. There are other ways to do this as well, but this is one of the more popular ones because he made a bunch of constructs and put them on Adgene that you could just use as is. But it's not as modulate. Like if you want to put your own perfect transgene in here, it's going to require some more tricky cloning than just plopping in a cassette into something that has a simpler design. That being said, it's often other forms of intersectionality are much more common these days. And one is intersecting, say, your floxed allele with, say, infecting an axon, which allows retrograde transfer. And retrograde infection is actually very important for neuroscience, where you can infect, let's say you want to infect this striatum here and it projects to the SNR, then you inject into the SNR and you're labeling the cell bodies here because it's labeling the axons. Some serotypes do this better than others, as you can see in this quantification here. And in particular, one serotype was engineered through mutagenesis screens to actually do this very well, where you can see here where they injected this particular nucleus, where you can see that the red infection from their particular retro AAV by far outpaces the green infection, even though that's a little bit cheating because the red is always going to look much brighter than the green, but it's a very powerful tool, but many other serotypes do this as well. Now, another thing that in neuroscience is particularly used in certain fields very extensively is activity-inducible AAVs, where you can have, in this particular example, a minimal promoter for CFOS, an immediate early gene that turns the TTA, which in the absence of doxycycline, binds to this TRE, this tetracycline response element, and then expresses this. This is a form of intersection where you have particular cells that are active, that are active in the presence, that due to their activity, they turn on TTA, but then also in the absence of doxycycline, that TTA is active to then turn on mCherry. This is from an example with the so-called engram cells in the MPFC, which allow for you to tag the cells that are active in one day, presumably during one particular behavior. But this is, of course, subject to interpretation, but is a very powerful form of intersection. And it, of course, depends on if your system likes CFOS. But this is also applicable to all sorts of other things. Like you can use a developmental gene that has a Cre line and then inject just DIO there, and then you'll get an intersection of your time of injection and that developmental trajectory as well. Now, you don't have to fill in those 4.0 kb. In fact, the shorter your construct, the better your viral titers are going to be in general. And in fact, if it's floxed, you usually get better viral titers than if it's unfloxed, because when it's in the 293 T cells, you're not producing all that RNA and protein. So the system tends to be able to make viruses more. But there's actually one particular type of AAV, the self-complementing AAV, that allows for much faster expression in a variety of systems. And this is possible because one of the rate-limiting steps of the AAV expression is because it's single-stranded, is to produce the other strand and complement. So people have very cleverly mutated one of the ITRs in a way that the AAV actually gets packaged into the capsid as a doublet that then goes back into itself and serves as its own complement, just thus self-complementing. And as you can see here from this, from the paper that first described this strategy, that you can see the time course of the normal AAV and the one with the SC AAV, showing that you can see a dramatic increase in the speed at which you get substantial expression compared to the non-self-complementing. Of course, this is a very limiting thing to do because you're then restricted to a 2.2 kb, and that includes your your promoter, your poly-A, if you have a UTR, that adds up. So only if you have really, truly small constructs will this actually work out. And oftentimes, when you're doing that, you're expressing Cre, which is tiny, and you need very small amounts of it. So you don't usually need an scAAV, because even at very, very small amounts, it is actually performing the recombination event that you want it to do. But it's an option there that was probably going to be useful in a variety of systems. Now, CRISPR-Cas9 is all the hotness, and unfortunately, the Cas9 gene is really large. So people have been able to engineer variants where, in this particular study, for instance, called the viral slender, where you can deliver the Cas9 with a really minimal promoter and just the bare minimums of what you need here. Like, it's really reaching that limit. And then with a separate AAV, you have the HDR template and the sgRNA in order to do the knock-in that you want in vivo from a virus. And these are just some images of them knocking in into the CAMK2 locus, this HA tag here. And you should be able to use this in order to do in vivo imaging of a variety of different systems, especially if you don't want to perform this genetically in any way. There's also a newer variant of this, which allows for you to, which contains some minor improvements for certain aspects of this, but I'm going to basically skip this over. So one study has recently claimed that AAVs can also be transsynaptic and pterograde. So that means that if you infect a neuron, say neuron B, with an AAV expressing Cre in an animal that's TD reporter, you're not just going to get TD tomato expression on B, but also in C, which is downstream of that. Several caveats to this. They claim that it can only be done with a few serotypes and a few promoters, as shown here, with extremely high titer, and only if you express Cre in a reporter line. And you always see a very, very sparse amount of postsynaptic infection relative to the amount you infected. So like in this case, they infected the retina, and this is where the retina projects to in the brain, and one of the places it projects to, the LGN. And you can see a very, very, very dense plexus of these retinal inputs into the LGN, but only a spattering of cells, very dimly expressing the TD tomato. Now, this may be a real thing. I tried to replicate this, but in my hands it couldn't. And of course, it could be that the serotypes weren't right, and I didn't try all the the best titer maybe or anything like that. But I've seen in several studies that when people use this, they often underestimate how much, for instance, C would project back to B, and that you would then get retrograde labeling, where a lot of the thing you think is anterograde is actually just you're getting retrograde labeling, where you're actually labeling the axons from C that project to B. Because if you're doing extremely high titer virus, almost every and you're waiting for a very long time, almost every serotype is transsynaptic, as just a way that things kind of go. Now, so how do you engineer viruses? This is now cutting edge of what people are trying to do to make viruses do some interesting things in their systems. And people have been trying to make mutations in order to get AAVs to express faster. And there are a lot of rate limiting steps into what an AAV can or can't do, such as when it can actually leave the endosome, when it can actually do its things in the nucleus. I mean, this 4-week thing is ridiculous. I mean, it makes no sense from what we wanted to do. But apparently that's been not yielding something that is particularly useful for tool users. And people have also been trying to use predictions as to what certain domains of the VP cap proteins in particular are doing in order to see if you could get serotypes that are specific to a cell type, like just an excitatory cell or just a liver cell AAV or something like that, with not really very robust results. What have led to robust results is capsid screening in that regard, whereas people have been able to develop a photoreceptor specific AAV that when you infect the retina, it used to be that you just couldn't get photoreceptors very well infected. And but then this one came along. The retro AAV that I talked about before was done by taking a capsid and just mutagenizing it, and then seeing and selecting for ones that allowed for larger retrograde pickup, as well as the systemic AAV. And this is what I'm going to talk about in a little bit more detail, just to introduce the concept, because it has been so powerful, and it's been very powerful in a variety of different systems. So you basically have an AAV that expresses your cap protein. So you only have to grow it, you only need to provide it with the rep and the adeno proteins. And then you have it in such a way, if this is necessarily not a part of it, you can have it pre-inducible, but you basically just make it such that each viral construct has a different cap part of it, like a diversified region that is mutagenized. Then you infect it in vivo, and then you select from the cell types that you want, and you sequence out what are the ones that are required for this. And the Gradinaru group has been able to take AAV9 that at very low titer, that, you know, at reasonably high titers does cross a blood-brain barrier, but not very well. And through inserting a heptamer into one of the sites in the VP proteins, it's one protein with different sizes, they were able to mutagenize it so that it created a variant that was much better at that. And then they mutagenized that further and made it work even better by just a 2 amino acid change to amino acids just flanking that heptamer, which is quite potent. So it seems like this domain here is very important for the virus infecting cells. So it also really limits what you need to mutagenize in order to get different cell types, at least a priori. I don't know how this would reflect in systems that are not neural, but I believe these other ones did not use dramatically different domains as well. Now, the virus efficiency in terms of how it affects different systems is not just the capsid, it's also the genome inside. You have a promoter in there. Now, the problem with all viral promoters is that they have to have a limited size. They're short. You know, and in vivo, you have a gene that has regulatory elements in the introns. They have enhancers and suppressors like crazy far away in the genome that through genome folding can go into the promoter and affect transcription. Whereas, so if you want to put that into max 2 kb that you'd realistically fit into an AAV, and even then 2 kb is a bit much if you want to put in all the bells and whistles that you'd like. So the Roska group where I did my postdoc had this project where they actually tried to make artificial promoters based on a variety of different designs. And they did it not as in the previous screen where they just infected a lot and then selected and sequenced. They actually did it the hard way where they injected in vivo with a catch GFP construct in a variety of these synthetic promoters and just assayed to see which ones had the good expression. Now, this is interesting because then they tested over 500 constructs over several years, which was a lot of labor. But with this, you could actually, it kind of underlines how little we know about these regulatory elements. Like you can see here that we took, you know, the binding sites for transcription factors, you took conserved elements upstream of genes, the 2 kb region upstream of genes, methylation-free areas upstream of genes, and the output that you got sometimes was incredibly clean, but not at all related to what we picked it out for. Like this, particularly this cone-specific promoter, was not from a cone gene, it was from a rod gene. And a lot of these have this kind of thing where it just sort of makes sense, but not really, which just underlines that the knowledge that we have in order to design promoters is not really there. So either you design a screen to screen through a whole bunch of things and do unbiased mutations, or you have to really just try 500 things in order to get 30 to 50 to kind of work. And I would not recommend any single person do that, by the way. So that's it for AAVs. Anyone have questions? Take a little breather break. I can drink some tea and chat. Anyone in the chat? All right, good. So, okay, so now we're moving to lentiviruses. This is an older virus. Perhaps it's the one that dramatically kicked off a lot of interest in virology because of the HIV crisis, which in a very similar way to our own now was incredibly overlooked for a long time, which caused a lot of suffering. But it also caused a lot of interest and study in it, making it perhaps one of the first tools that were seriously used in order for gene manipulation in mammalian systems. So it encodes for 3 particular genes between these long terminal repeats, the GAG, the POL, and the ENV. And I'm going to skip a lot of the details of this because it just, the important thing to know is that it has a capsid, but around it also an envelope. And the envelope is different than a capsid. These, the icosahedral capsid from the AAV is just a series of proteins that together form this shell. An envelope is actually a plasma membrane derived from the cell that the virus infected. So it has different properties that will become very important. It has a whole bunch of other things as well, such as an integrase, which allows for this to be integrated into the genome. It has a reverse transcriptase that turns this RNA virus into DNA, which has been incredibly useful for molecular biology in general, and going with a theme of viral discoveries being useful in ways that we did not really predict. And when you grow this virus for use, you basically remove all these genes and you add your promoter and transgene and all the constructs and little bells and whistles that we mentioned before. And when you produce this, I'm not going to go into the details. It's slightly more complicated because you need to introduce a lot more genes in order for it to be packaged properly. It basically comes to a decision if you want to do second generation or third generation lentiviral production. Third generation is safer, but it has one more plasmid, which makes it a bit harder to get high titers if you didn't optimize your transfection as well. And I'll leave it at that. It's a pretty turnkey technology at this point. They are also Cre-inducible in a very similar way as the AAVs are. There was earlier some difficulty in getting these viruses to be Cre-inducible as well as AAVs because people were growing them in cells that allowed for recombination. So you'd actually get recombination or even recombination within LTRs or ITRs because they are repetitive domains. So, but nowadays everyone knows to grow these things in bacteria that do not allow for recombination. And in fact, most labs that are viral as a policy just use those bacteria because they don't want their viral constructs to suddenly stop working. Used to be you had to check for that, and you could still do that as a sanity check if things are not working well. Now, lentiviruses have limited spread, as I mentioned last time in this particular study, which I want to talk a little bit more, because it also illustrates that it has this much higher cargo capacity of 10 kb. Where you can see here in this study, they had a CAMK2 promoter, which is not small, expressing GFP, and then an internal ribosomal entry site, which again is not small, and doesn't work as well in AAVs as it does in lentis and with making mice. The 2A construct is a lot more powerful in the AAVs, and then it expresses Cre. It's not the only thing. There's all these other bits here as well, which I'm not going to talk about. But this particular study used it to actually do inject this virus into a single barrel in the mouse primary somatosensory cortex and deliver Cre, which would then knock out the NMDAR receptor while maintaining the other nearby barrels intact. And this is a thing that this particular construct would excel at, which is this localized infection here, while still delivering a pretty decent amount of construct and gene expression. However, for the vast majority of applications that people use, you kind of don't want that. You want a large bolus of your tissue to be infected, and you actually want to use these intersectional techniques that we mentioned before to actually then limit the infection to something functional. Like let's say you want to inject into a brain area that say projects to region X, but also are parvalbumin positive. So you take a PV Cre mouse and you inject over here. That way, in this area here, you're only going to get the PV positive cells that project there, expressing your transgene. And this is important because then you're not going to label all the stuff nearby that either doesn't project there or does not express PV. So it turns out to be much cleaner. And you can also do a much bigger, robust injection to really infect as much of that structure as you can. And you can easily think of using these intersectional techniques for a variety of different applications in other fields of biology as well, from in vitro to other organ systems to whatever. Now, an important part of lentiviral use is that you can pseudotype these viruses. And this is because it's enveloped. So instead of using the normal lentivirus envelope, you can use something like the vesicular stomatitis virus glycoprotein envelope, which infects then only the cell bodies. You can use a murine leukemia virus glycoprotein. You can use the rabies glycoprotein, which allows for pretty robust retrotransposon, or a rabies VSV chimera that works a bit better. But most potently, you can use the NVA system. The NVA and the NVA ligand and TVA receptor is actually the first ligand receptor pair studied in biology, and is an incredibly potent pair that exists only in chickens and not in mammals. I don't know below chickens if like reptiles or whatever has it. But the point is that if you have your lentivirus enveloped with envelope A, for instance, you can then express the TVA receptor from a variety of different techniques, either with an AAV, with a transgenic line or whatever, a knock-in or something, to then make sure that that lentivirus will very specifically infect that cell type. And this is exemplified by the study where they have different variants, the envelope A, B, or E expressing different fluorophores, and they separately, and they inject retrogradely, so they infect the axons of this neuron with a vector expressing the TVA receptor, and then in that nucleus, they mix up these vectors that express these lentiviruses that each one has a different ENV-A-coded construct. And you can see here that you see zero expression of the ENV-B or ENV-E, but pretty robust one of the ENV-A. And so this is actually one of the cleanest ways you can get very specific infection. So, you know, this is a goodie. Now, you can also do cell type enriched promoters. I would not even call them cell type specific, because it's kind of like a broad classes of specificity. It's not like the ones that they picked out from the promoter screen for AAV. These can be called that they're in broad classes, but if you look at the details, they're not highly specific. It's more of like a strong bias towards one cell class or the other, but people have found that useful still, because oftentimes, you know, you don't care if something is 95% of excitatory cell or 99.9% of excitatory cells. It'll be good enough for what you're doing, at least in the past. Recently, there's been some movement in terms of making lentiviruses integrase-free, so they would not integrate into the genome. And this wouldn't be super necessary for basic research, but would be very important for gene therapy and vaccinations. And in particular, there was one paper that found that if you can express genes for much longer, much stabler in terms of producing a vaccine in the system than you could with a more traditional approach. We'll be talking a bit more later about why the integration of the lentivirus is a particular problem for clinical applications. And however, this ability for the virus to integrate into the genome allows for something called lineage tracing, where you can actually infect cells that in development, which then you can then see their progeny. And I'm going to skip the details of this because I briefly touched on it last time. And it's something that you could say look up in this sort of paper because it's kind of fringe, and I'm kind of going a bit over time. I'm sorry, this is not predicted. Now, an important actually application for lentiviruses is that it's a very elegant way of doing ex vivo gene expression CRISPR screens. Because you can pack so much into it, you can put in the Cas9, you can put the guide, you can put all sorts of crap in there, like a pyrimycine for selection. And compared to the old shRNA, which had all this variability and never really a full knockout effect, different guide RNAs can really clench that thing all the way down, which allows you to really be able to produce a library of lentiviruses targeting different genes, which will then have knockouts and then produce your functional screen in vitro, which would then allow you to select genes which a lot more confidence and less variability than with an older shRNA screen. Now, this is something that really drove a lot of how people think about using viruses in basic biology and its application into gene therapy. There's this disease called X-linked severe combined immunodeficiency, which means that with one gene mutation, you have no T cells and no NK cells. Like your immune system, your adaptive immune system is busted, meaning that you have to really live in such a way that is prevent any possible type of infection from bacteria, from fungus, for anything. And it's actually very difficult to do. And one of the few ways to treat it is if you could get a bone marrow transplant before 3 months of age from someone you have an HLA compatibility, which is often not really the case that you can get. But it's a prime candidate for the gene therapy because you only need to replace that one gene in the cells that you can extract from the bone marrow and then put right back in. And will then be amplified in the patient. So they did this with 9 patients that couldn't get a donor. So really had difficulties in their day-to-day life, and generally have a much reduced lifespan because of just the practical impossibility of living with this disease. And so their bone marrow cells were removed, a lentivirus carrying this gene infected them, and then put back in. It looked like success. 8 of them had initially successful, where they detected T cells and NK cells, and 10 years after the gene therapy, they were still producing T cells. And it corrected the immunodeficiency, basically, like they were normal. The problem is 4 of these patients got acute leukemia, and 1 of them died of this disease. Now, 4 out of 9 is pretty terrible. And this is what actually really underscored the problem that you could have a lentivirus integrating into a gene region that is highly activated, and that make it even more highly activated, and then turn it into an oncogene and thus cause cancer, which is what happened in this case. Which is why for the longest time, for over 10 years now, people have been extremely hesitant of applying lentiviral anything to the clinic because of this experience here. So this is why people talk a lot about this particular issue of integration and why AAVs became a thing. Okay, good. Question. Dimitri asks, are there examples of increasing specificity for AAVs by using ligand receptor pairs similar to Lenti-AAV strategy? Yes, actually, Paul Heppenstall, who recently was a group leader here at EMBL Rome, is devising such strategies for infecting very hard to infect peripheral tissues. But that's still not published yet. And the short version is that it's very difficult because you start adding big things to the capsid and it starts falling apart. I mean, it's like a very regular, beautiful little platonic thing, platonic solid. So if you start messing around with the proteins there, the capsid can often not form very well. Or in his case, often using like chemical additions to it and all sorts of tricks like that. But as far as I know, people trying to do that haven't, and you can't use the actual NVA TVA system because the protein is too big. If you fuse that to the cap proteins, things apparently don't work out. Good. Are there any other questions while I take a drink? Good. I mean, you can't do something like that, but there is a lot of promise in doing these capsid screens where you can mutagenize the capsid and then screen for what you want. Of course, the ones that I know have been done in neuro circuits, so beyond that, it remains unclear to be seen how effective they will be. Right, so rabies virus. Very different kind of virus. RNA virus, and it's negatively stranded RNA. So it can't just start pumping out protein as soon as it gets into the cell. It needs, for their own polymerase, the L protein that is packaged together with it to actually start producing each of these RNAs separately. And it produces the N, then the P, then the M, then the G, then the L. And there are these little transcriptional stops and starts. And whenever it stops one set of transcription, there's a chance the L will just fall off and start over, or it'll continue to the next one, which ends up producing this transcription gradient where you have more of this and less of that, which is important for the packaging of things in general, where you need very little of the L, but you need a whole lot of the N to form the nucleocapsid, as well as a lot more of the P to put it all in there, the matrix, and so on and so forth. But what we really do when we use this is that the glycoprotein, which is very essential for the virus to then be translocated to the outside of the cell and then infect the cell it's going to infect next, you replace that with something, say, like GCAMP or GFP or whatever. And then you grow it such that the virus itself has all of this, all of the stuff that the virus itself has all the things that it needs. It has the G protein on the outside, it has its genome, but once it infects a cell, it can't jump anywhere because it has no G. It can start pumping up a lot of expression of the other proteins, including the GCAMP, but it can't jump. So what you do is you transcomplement with a G protein, where you have this G-coded infection, but you also allow expression of the G in a separate say. So if you say have this daisy chain of neurons A to B to C, you infect C with this rabies virus that is G-coded and expressing GCAMP, but you also infect it with an AAV expressing this particular variant of the G protein, which allows for much more robust transcomplementation. you'll not just get infection of C, but you'll also get infection of B. Oops. You'll also get infection of B, but it won't jump further to A because the B cell is not expressing this G protein. It'll only be expressing all these components in here. And for instance, this was used by Sylvia Arber's group to infect motor neuron cells and then jump to the cells in the spinal cord locally as well as further away in the brain that connect directly to these chat-positive motor neurons. And this is particularly powerful because it jumps after one synapse. So you don't have to wonder like, oh, is this a direct connection or 2 synapses away or so forth. The problem with this is that this rabies G, as I mentioned before when I was talking about pseudotyping, is very good at labeling axons. I mean, that's what they did here, right? They labeled the motor neurons all the way at the muscle. So if you have, like, say, B also producing axons in this area here, you'll label that as well, which is a problem. But that's where the Envey TVA system comes in. If when you're delivering something to C, you're not only given the G, but also the TVA in, say, a way that you make sure it restricted to cell type C, then your rabies virus can be coated with envelope A, not the G protein. So it'll not infect anything else in your mouse or even in the human, but it will infect that cell. So people did this, say, in the barrel cortex, where they had a PV Cree mouse, where they infected with an AAV expressing TVA and the G, and they see rabies in all sorts of different parts of the brain. And if they infect just a TVA, which allowing just the cell to enter, they can see that you don't get direct infection of these brain regions because, again, you're doing an intersection between what your Cree line is expressing and where you're injecting. And it is unfortunate that we don't have this particular tool for the AAV, but there are other ways to go about it, like we talked about before. Now, another particularly powerful use of the rabies transsynaptic tracing is that you can start it from even a single neuron, where you can actually electroporate this DNA construct into a single cell in C and then infect the whole region nearby with an envelope A-coated rabies. And the TVA envelope A receptor ligand pair is so potent that works. Not only does that work, you can electroporate and inject that virus in the same day, and it'll take like 2 or 3 days for the DNA construct to express, and the virus will still be sitting around in enough quantities to actually infect robustly cell C, which is just crazy if you think about it. And so in this particular paper, again from the lab that I did postdoc in, they performed single cell electroporation of a pyramidal cell in layer 2-3 of visual cortex with constructs expressing G, expressing GCAMP, expressing TVA, and then they injected locally nearby the envelope A rabies expressing GCAMP so that they can then later look at all the neurons and perform calcium imaging in vivo of the neurons that are presynaptic to a single cell to look at what are the circuit components connected to a single cell and what can you tell about the visual circuit using that particular access to this monosynaptic circuit. Now, the problem with rabies is that it's very toxic. That starter cell that you see here, very rarely do you see your starter cell survive long enough to see it together with your presynaptic cells, which is a big problem if you want to image them at the same time. So people are trying to devise ways where rabies viruses are becoming less toxic. One of them is an L-deleted virus where you can see the G replaced with a Cree, and you have no L, meaning you don't get that amplification. You just get this virus entering cells and then just expressing a little bit, which is why it has to be Cree, because you don't get amplification with the L protein really pumping up the translatable RNA. And so far, the Wickersham lab, Ian Wickersham and Ed Calloway being the developers of the rabies technique, and Wickersham is now going on to further develop this L-deleted one as well. They've published it as just infecting neurons, not transcomplemented and jumping yet, but I'm sure that's on the works. And recently, another group has found that they've developed a rabies virus that once it jumps, and even before that, it starts being degraded by the proteasome with this pest sequence tagged to the N protein. And there's some debate as to whether this works as intended. Wickersham's group has reported that they found that vials of this virus have had this domain deleted out, because when you grow rabies virus in vitro over many passages, which is what you do when you grow rabies virus, you're effectively selecting against this particular domain, because without it, the virus will just be able to grow much more efficiently. And of course, the group that developed this disagrees, but it remains to be seen how well this particular reagent works. So there are other issues with rabies as well. And in fact, many very good neuroscientists and biologists in general have problems with rabies. A lot of them are very well exemplified in a blog post by Karel Svoboda, linked down here. You can take a snapshot now if you want to look at it later, or just Google Karel Svoboda rabies virus. It's a very long but detailed and rebuttal of the virus use. But his argument mostly has to do with the fact that the cells you see connected with rabies are not what you'd see from detailed circuit analysis, a lot of it using slice physiology. And so I think a lot of this has to do with downsides of rabies, which are well known, and some may or may not be unique to that system. And in particular, these are known issues with rabies that people should know before using it. One is that you can't use CRE with it. It's an RNA virus, so recombinases don't work. No cell types or promoters. It kills the starter cells, which is a big problem. You need to transcomplement very high levels. Like your AAV expressed in the TVA and G has to be a very good one. And usually this is a problem where people are not able to do that very well. Like their virus is not good, or their strategy is not labeling cells well enough, or so forth. But here's the big problem. These two are the biggies that often go to the heart of Karel and others' big problems with rabies. The first is that you have a false negative rate. Rabies likes to jump synapses, but not all synapses. And it is kind of unclear what the rabies likes and doesn't like. In the retina, they jump very well from ganglion cells to amacrine cells, but not at all from retinal ganglion cells to, say, bipolar cells. Like, I guess it doesn't like a ribbon synapse. So oftentimes you will not see synapses that should be there. So you should always bear that in mind. And another one is that it seems to be independent of how active that synapse really is. So it's just a binary measure of, are these things connected or not, is how people usually interpret rabies. Nothing to do with the strength of the synapse at all. And if you and this has been questioned by a few recent studies that found that, you know, with some cocaine addiction treatments or others, that you can seem to be a difference in the rabies jump, but I think those effects are not particularly robust, and I'm not entirely sure they go beyond the difference in batch you see when you often do these rabies experiments. And these two often really sometimes limit your want to really see the circuit in an unbiased way that actually reflects underlying physiology, which very clearly rabies seems not to do. So I'd like to finish with just showing on a very simple table a lot of the properties that I talked about before and which of the viruses have it or not. This table is not going to be that useful if you're actually deciding to use a virus or not, but it's here just as something to stone cap the talk. And I'd like to thank you all for coming. Yeah, so this lecture and the previous one was on these topics because they were the ones that when I originally sent out a survey asking about topics, they were the two by far the most popular ones that people really wanted to see. So the next step possible on this is very unclear. I might just end up doing something a little bit later on, you know, something that I find is kind of cool or whatever. But if you have any particular ideas, like there's room for that in the survey, and I very much appreciate input. But you can also just drop me an email or whatever. I'm on Twitter if you're not part of, you know, the circle that can get my email from the internet. Just, you know, drop me a line, and I'm always very happy to see about doing more of these maybe or whatever. All right, thank you all for coming. It was nice seeing you, and hope you have a good weekend. Bye-bye.